welcome back to the Probiotics Theater. Our next section of the day is Achieving Outcomes with Precision Microbiome Analysis and AI, presented by Blake Wills, the founding CEO of Microba, a cutting-edge gut microbiome analysis company which uses genomics to provide actionable insights. Following 4 years as Microba's founding CEO, Blake now acts on an advisory basis, focusing on key projects and alliances, which will impact the future of its customers globally. Without further ado, Blake, you have the floor.
Thank you, AJ, and thank you everyone for being here. Firstly, a little bit about Microba before we get into the content of the talk. The company was founded by two professors, Professor Phil Hugenholtz and Professor Gene Tyson. They're world-leading microbiologists in the field of genomics in particular. And after spending time at MIT, also the Joint Genome Institute and UC Berkeley, they moved back to Australia to set up a microbiome research foundation. That is the basis upon which we founded Microba some 5 years ago, and I'm excited about what we're going to present to you today. This is very straightforward for those in the audience, but clearly the microbiome has now been shown to have a significant impact in a broad range of health conditions. In fact, there's now over 200 studies showing that impacting the microbiome has outcomes on disease. So not just correlations, but actual outcomes. We know there's multiple levers in which we can affect the microbiome and in turn affect disease outcomes. We have prebiotics, we have foods, we have probiotics, and we have antibiotics as the main sources of interventions. Today I'm going to talk to you about how we use big data to identify the right tools to shift the microbiome. Firstly, though, some of the challenges when it does come to analysing the microbiome. We know it's a hugely complex ecosystem. We know that each individual has hundreds of species, obviously thousands more strains, but at a species level, hundreds of species, and then the functional genomic profile of each individual's gut microbiome is incredibly diverse. So understanding this is a huge analytical task, and that's where big data, AI, and machine learning really does come into play. So what have we done at Microba to tackle this problem? We've recognised that you need big data. So we now have accumulated what we believe the world-leading microbiome data set. So we have 15,000 gut metagenomes, so 15,000 samples, where we've got comprehensive metadata, so health data around disease, diet, medication, lifestyle, etc. But we also have what we believe is the best microbiome analysis suite, and we'll talk more about that in a moment. Importantly, if you combine those two factors, we believe that you can identify new product discovery opportunities. So quickly, in terms of analysing the microbiome, there are many ways to do it. You can try and grow things on a petri dish culture. You can use old sequencing technologies from the 90s, such as 16S RNA sequencing, or you can use the more next generation technologies. At Microba, we choose metagenomics, and we've now published on the superior nature of our metagenomics in contrast to other tools. And what happens if you don't use the right measurement technology? In short, this picture tries to illustrate that you'll miss an enormous part of the equation. Some of you may or may not know that 70% of the gut microbes have not yet been brought into pure culture. So if we're trying to use old techniques such as growing them or even 16S low resolution, we're going to be missing a very large part of the picture. And what we've learnt at Microba is that many of these uncultured organisms are the ones that actually drive impact and change when it comes to disease, health, and wellness. Just to give you one example, for those in the probiotics industry, you may have heard of the emerging strain Faecalibacterium prausnitzii. This is now broadly regarded as a very promising strain for probiotic use and live biotherapeutic use. Often in shorthand, we just consider Faecalibacterium prausnitzii to be a single species. But when we actually look at it through a metagenomic lens, using the leading taxonomic profiles and measurement tools, there's 27 individual species of Faecalibacterium prausnitzii. I'm not talking about strains, I'm talking about species, some of which are still uncultured and can only be measured genomically. And I'll get to this later in the talk, but if you have a strain of Faecalibacterium prausnitzii, it may not be the right strain for impacting the disease or the health outcome that you're trying to. So it's really important that we have this high resolution data when we're trying to identify new product opportunities. So back to our data set. So this 15,000 microbiome data set actually has a lot of disease signals in it. I've listed a few of them there, such as irritable bowel syndrome, rhinitis, also anxiety. And we'll use anxiety as a bit of a case study today as how you can go from big data with anxiety through to new product opportunities. And as I mentioned earlier, there's also very rich metadata, which is important in the microbiome because a lot of these metadata points are confounding factors. So if you've got different diet, lifestyle, gender, age, etc., you can actually remove that noise and actually get to the true microbiome signal. Just to demonstrate the role the microbiome is playing, here you'll see 8 indications and 2 different coloured graphs. One is orange, which represents the disease state, and the other is blue, which is our healthy cohort. And using an AI, so this is a deep learning neural network, we're able to clearly separate the signal of a healthy microbiome to that of the indication. And within that is product discovery opportunities. We can identify what is the difference between the disease and the healthy cohort, and we can see if there's some key drivers in there that'll actually let us try and treat these respective conditions, whether they be in a disease setting or more of a health and wellness setting. So what does that look like in practice? We start with a big population data set. We clearly separate the healthy cohort from the disease cohort. We use AI and advanced biostatistics, including cohort matching, and as I mentioned, deep learning neural networks. And from that, we're able to identify a list of leads that are available for further testing. One of the exemplars of this we've done actually in inflammatory bowel disease. And here on the right-hand side, you'll see a phylogenetic tree of a particular family. And there's 2 dots represented for each species. The grey dot is the prevalence of that species of bacteria in the disease state, and the green dot is the prevalence of that bacteria in the healthy group. And you can see that very clearly different species have different prevalences in health versus disease. At Microba, we're actually progressing one of these into a clinical trial this year. That candidate's called MH27, and you can see that it's highlighted on the slide. It has clear separation between those within the healthy condition and those with the disease state. And in terms of the translational outcome that achieves by taking this big data approach, you can see on the histology there in the top left is what healthy looks like. On the bottom right is what it looks like after both receiving the disease, but then our treatment. And you can see we achieve epithelial restitution, crypt reformation, and it's a big part of what we believe is going to be the future of inflammatory bowel disease treatment. We see in context of inflammation and also epithelial injury, the histopathology scores are very high. Now, I just wanted to use that as an example to show that this big data approach actually does translate into real outcomes. So back to the anxiety example. This talks not about the gut-brain axis, but I did want to obviously share that the gut-brain axis is now being well established as a mechanism by which conditions such as depression and anxiety are being impacted. We know that there's clearly immune modulation, we know the vagal nerves included, and also hormonal signalling. I apologise, this clicker is running a little slow. In terms of the cohort that we have, as you can see, as per the DAS index, which is the validated survey for depression and anxiety, we have a very large number of people within our database that actually have anxiety, and then we have a really strong healthy cohort. So this lets us actually tease out the difference between health and disease when we match it up. How do we go about doing that? Well, we have our data set, we identify the confounding factors, and then we identify the taxonomic profile, so the species that exist within the healthy cohort and then the species that exist within the anxiety cohort. We match the dietary data, and then finally we look at the functional profiles. And it is important to look at function here, not just taxonomy. So looking at differentially abundant pathways is a clear way to actually getting to what is driving the difference between health and disease. This is well published on, so I'm not going to blow you away to tell you that anxiety, if you have anxiety, you have a lower level of diversity. That's replicated within our results. But what is interesting on the right-hand side here, we can see again applying an AI model, it's a deep learning neural network, we can actually compare health and anxiety and predict based on your gut microbiome alone whether or not you will have anxiety per the DAS score with an area under the curve of 0.88. So in other words, it's very, very predictive based on the gut microbiome whether or not you're going to have high or severe anxiety. That gives us great hope that there's an opportunity here to therapeutically intervene and actually drive a new product that will resolve anxiety. So, as I mentioned, there's a number of ways in which we can intervene. The first one is probiotics, and that's what we're here to talk about today, being on the probiotic stage, but I will also go through some other examples. So, there are 160 species in total from our analysis that are strongly correlated or associated with anxiety, and this is after statistical correction. And here on the plot on the left-hand side, we can see the top 10 species. And you can see the blue bar represents the prevalence of the species in a healthy person, and the gold bar represents the prevalence of the species in the anxiety. And you can see clear differences in the species abundance or prevalence when you're actually contrasting these two. What is also interesting, you'll see species 1, 2, and many of the others are actually increased level of abundance within healthy and decreased in anxiety. But you also get the reverse. You get some species that are overrepresented in anxiety, and therefore they could be targeted. You could try and downregulate those species. So there's the opportunity to upregulate or downregulate depending on what your product strategy is. Beforehand, I showed you the AI model that actually generated a prediction of anxiety with 0.88. On the right-hand side here, we can see a plot which shows what are the key drivers or features within the AI model that contribute to separating health versus disease. And you can see there's some really nice separation, both in terms of species and also function. We can also look at this in a network context. So you can see the blue dots here represent healthy, and the red dots represent anxiety. And what we see is there's a separation between the two, but when you actually look at it, there's also some keystone species. That's a concept that's been thrown around a bit lately. There's some species that are core and related to the existence of other species that are either pro in terms of reducing anxiety or negative in terms of they make the condition worse. Now I want to drill into one particular species or how you can use this data. Well, first of all, you can go through this list of 160 species that are strongly associated, and you can say, well, are there existing probiotics that are already commercially available that we can now take and apply to anxiety with this great big data that we have to hand? So that's the first thing you can do with it. The second thing is you can look at other species that aren't currently probiotics and look at new product development. So you could look at a new species of bacteria and say, this is really strongly associated with health and absent in anxiety, and let's develop this as a probiotic. And then finally, you could choose to take a prebiotic approach and try and upregulate good species based on what they grow on and what they thrive on. So there's three ways in which you can use this data. Coming back to Faecalibacterium prausnitzii, which I mentioned earlier, one of the top species in this is actually Faecalibacterium prausnitzii, but it's not one of the well-known Faecalibacterium prausnitzii. So if you just have a general signal and you think that Faecalibacterium prausnitzii in general is relevant, I can tell you that only one of the 27 Faecalibacterium prausnitzii are actually relevant. So this high level of resolution is really key to getting to the data. So let's talk now about the opportunity to develop novel probiotics. I want to take species 4, which you can see is highlighted with a purple bar here. You can see there's a very different effect size or very large effect size in terms of the prevalence of that bacteria within the healthy group versus the anxiety group. Importantly, it's also a Keystone community member, so it's positively associated with a lot of bacteria that also have a positive impact on anxiety. We know how to genomically analyze this, so we can look at the safety profile of this bacteria, and it's very good in that regard. It doesn't contain any antimicrobial resistance genes or other things that we might find concerning. And based on the genome, we also know how to grow this bug. We can look at how we selectively apply hammers that actually will upregulate the growth of this bug. So it actually presents an opportunity for the development of a novel probiotic, species 4 here. In addition to taking a probiotic approach, clearly we can take a nutritional approach. I'm going to stick with the same species, so species 1 to 10 there are the same from the earlier graph. And if we focus on species 4 there, we can actually look at food items that upregulate species 4. So we know species 4 is good in an anxiety context, and we know that there's certain foods, individual foods, that are statistically significant in upregulating species 4. Now, what's more interesting, if you go from univariate to multivariate, you can actually get a signature of food. So you can get multiple foods together that explain the upregulation of species 4 within someone's gut microbiome. And often it's not a linear approach, so when you apply multivariate analysis to this, you're going to identify the key drivers of the upregulation of species 4, which is what we're trying to get to solve anxiety. Again, as I mentioned, you can do this in a non-linear fashion, so what often happens here is that it's not as simple as you want to add more of food or ingredient X, because there may be a limit to how much food item or ingredient you actually want to give, and at some point it will actually become too much. So you can apply machine learning to identify exactly how much of this ingredient you want before it becomes bad. In addition to nutrition, there's the opportunities to look at polyphenols. We've done a lot of work in this space as well. So if we look at polyphenols, again, sticking with species 4, we can actually look at individual polyphenols that upregulate species 4. I'm just sticking with species 4 here as the example. Clearly, you could do this with any bacteria. Again, you can identify singular polyphenols that will drive the growth of species 4, or you can identify a combination which will significantly explain the abundance of species 4. So there is an opportunity here to take either a probiotic, a food, or a polyphenol approach. So a little bit of a summary here in the context of the polyphenols. We have great predictive power in terms of the microbiome's role in the context of anxiety. We know that there's 218 polyphenols that are negatively associated with anxiety. So there's a great discovery opportunity or data set from which we can work. And we know that the polyphenol signature, if we do it in a multivariate approach, can actually explain a big part of the microbiome's role in anxiety. So you combine all of these factors and you get a very strong opportunity to tackle anxiety with new products. Just like to make a few points in summary. The microbiome is still at its very early stages. Clearly, we're discovering new bacteria. As I mentioned earlier, 70% of bacteria have not been brought into pure culture. So whilst there's an enormous amount of probiotics that are used globally, we still are likely not working with some of the best ones. In fact, I'm very confident in that statement. We know that from our data, most of the conventional probiotics are not regularly seen. So we do need to start working with endogenous microbes, and those microbes we can identify from the data. Not only can we identify them, but we can also work out how we isolate them using their genome. It's not only about the taxonomic profile, but it's also about the functional profile. And using that same functional profile, we can not only isolate them, but then once we get them and dose it, we can look at symbiotic strategies or prebiotics that will actually upregulate their growth once they're dosed, so they actually survive in the system. It is a very complex system, so the use of AI and machine learning does come in handy, but you don't want it to be an entire black box. It's very good for identifying signatures and identifying which features are driving it, but you do need to actually come up with a lead list and then go through traditional testing processes. But if you can start with a lead list of 10 or 20 very robust leads instead of hundreds, you're going to be in a much better spot. That's all I had today. I hope I've convinced you of the role of big data, the microbiome, and the need for advanced statistical approaches to measuring it. At Microba, we are open to partnering as well. We have built this data set. We're not experts in building new products. We're experts in microbiome analysis, big data, and identifying these opportunities. So if there is an interest, please do come and talk to myself or Drew if you put your hand up in the audience there. We'd both be very happy to talk to you as how we might work together. Just a couple of quick acknowledgements and my contact details. So that's my contact details on the screen there, my email address. So I'll give you a second on that before making a couple of additional acknowledgements. As I mentioned earlier, this company was founded by Professor Phil Hugenholtz and Professor Gene Tyson. Their ongoing involvement in this company is instrumental, so I'd like to thank them for all of their work. But we do now have a team of 60 scientists that are contributing to the discovery of new microbes, new product opportunities, and if we can work together on this, we'd love to. Thank you very much.
Thank you, Blake, for joining us today and sharing those insights with us. A reminder to our audience, if you do miss a session today, you can always catch up on demand on the VitaFoods Europe online platform. We will now open up the floor for some questions, and we do actually have one that's come in from online. The question is, how much criteria can be applied for a regular diagnosis of ADHD?
So I'm not entirely sure if we have looked at ADHD, or I personally haven't. I'm sure someone in the science team has. But what we are seeing, as you saw on those graphs, is there is strong signals for a lot of conditions coming from the microbiome. So it would be worth looking at. I dare say we would have some very interesting data in that regard. I think it would likely be diagnostic, but we'd have to go and do the exercise.
Sorry, do you have a question?
Hi, can you do the same with a prebiotic? For example, can you follow it right through the microbiome and see what effects does it have in short-chain fatty acids or neurotransmitters in the subjects of the study?
Absolutely, yes. So what we do, and we have an arm of our business that actually supports this. So if you want to run a research trial or a clinical trial, we can analyse the microbiome and what changes have occurred to the microbiome. That's both at a taxonomic level, so what species changes are actually occurring, but also at a functional level. And when you talk about short-chain fatty acids, we can measure the genes and the change in the genes as you dose the prebiotic. So we can do a pre and post, and it's a very good idea if you're trying to prove the efficacy of a prebiotic that you might be developing.
Thank you.
Thanks very much for the talk, Blake. Ian Brownlee, Northumbria University. You talked a little bit about food and food signatures there, and I suppose your systems and the power of your systems is built on the microbiomic analysis. It struck me there as how do you, do you look at the data in a different lens, the relationship between food intake, which is not quite as complex a data set as your microbiomic data, but it's difficult and it's awkward to handle. You've got whole foods, you've got food groups, you've got food components and subcomponents, as you hinted at there. Have you looked at the healthy and disease state populations only based on food and not through that sort of starting point of the microbiome and things that can change the microbiome and then go in that way? Because I guess if you're looking at a food-based intervention, microbiome is certainly a potential mechanism of action, but could also just be affected by dietary intake directly or indirectly based on, even just something that goes through the gut quicker and changes transit time. I think there's some strong relationship there now with microbial changes and outcomes there. So, you start off with the microbe, and sometimes when I talk to my colleagues in microbiomics, they always start with the microbe, and are you looking at it from different points in terms of the wider data set that you have access to there as well?
That's a good question. We certainly have the data points, but it's not our focus as a company, but we definitely have the data to say, well, here's a healthy cohort and here's their diet versus here's a anxiety cohort and here's their diet. So that opportunity is there. And I think your point is sound. Not everything is going to be causal. Some of it is going to be associative. And it's about identifying which ones are causal and which ones are associative. So it's a very sound point, and the opportunity is to do that analysis as well.
what you were talking about there, a microbe-based intervention absolutely makes sense though as well. It's just, yeah, sort of in my mind, that separation between the two bits could be useful, and could be another use of your sort of AI approach and existing data set as well, I guess, because you don't always need every intervention to be dependent on microbial change, I guess, as well in a human.
Yeah, it is a great point. Our company has founded around the microbiome being mechanistically relevant, so that's hence our focus on the microbiome, but yeah, you could certainly look at it without the microbiome as well.